Can plants learn? Phototropism Lab. Introduction. The problem of the experiment was whether or not plants learn, along with how plants' growth is affected by phototropism. As plants are unable to relocate themselves, they must be responsive to whether changes happen in their environment. One such example is a plant's response towards their light source, also known as phototropism. This process occurs with the plant sensing the direction, quantity, and color of the light source. Many plants, such as sunflowers, heads follow the sun, and legumes lower leaves in the evening and raise them in the morning, are responsive to light cues. This cycle of response is called circadian rhythms, and they are around 24 hours long, coordinating with the positioning of light, in some sense creating a biological clock. The hormone that was responsible for the growth was auxin. Auxin is a plant hormone which causes the elongation of cells in shoots and is involved in regulating plant growth. This occurs when the auxin molecules are on the outer wall layer of the plant and bend towards the light source, increasing the efficiency of absorbing light. Hormones control plant growth and the development through their ability to affect division, elongation, and differentiation of cells. This is important as the appropriate response to light is crucial for a plant's success. Light signals plants' growth and development. Problem: can plants learn? In a conventional sense, plants do not learn in the same way that humans do. As they lack a nervous system, this doesn't mean their ability to learn is determined by the process of the brain. In this video, we will explore how and why behind plant growth due to their exposure to a light source. Additionally, we will determine if plants are able to learn by identifying hormones responsible for behavior, being measured along with connection to our work in the podcast Smarty Pants and From Tree to Shining Tree. Plants use a variety of hormones to influence their growth through these processes. The first option is phototropism, which is growth in relation to a light source. The next is zygotropism, which is growth in relation to touch. And the last is gravitropism, which is growth in relation to gravity.
For the personal engagement, movement is a fundamental aspect of life. As plants are anchored and cannot pick up their roots and move when they are not in optimal environment, it is important to understand the conditioning for movement. Movement in plants is a result of growth as a response to various stimuli. For example, plants will grow towards light and their roots will grow towards water, as a way to increase their ability to obtain resources. This response is complementary to the finding of psychologist Ivan Pavlov in his work in classical conditioning. In Pavlov's experiment, he explored classical conditioning of dogs for the subconscious associations of an unconditioned stimulus to an unconditioned response, ultimately creating a conditioned response. Plants don't necessarily learn in the same sense as humans or dogs, but they do have signal transduction pathways that link the signal reception to a response. Plants have receptors to help detect any changes in their environment. This response to light is an example of cell signal processing. In this image, you can see a cell signal processing. A signal transduction in plants almost parallel to the understanding of human response to a stimuli. Plants are one in the same growth. Growth responses are usually slow and are not reversible for both humans and plants. By understanding the impact of conditioning and stimuli, one can shape the outcome. Overall, outcome of the experiment shows us that the stimulated circadian rhythm and a sense of time of day is closely related and oftentimes compared to modulate behavioral processes, such as learning in animals. The experiment also shows us that the plants have associative learning and that plants not only respond to light and food to survive, but that they also choose and predict in order to survive. Associative learning is a concept where a new response becomes associated to a particular stimuli, such as the experiment with the dog. The term basically describes learning, which shows us how plants have far more sophisticated abilities than we previously thought.
Exploration. Research question. What is the effect of light on the growth of a plant? How do plants react to their circumstances as they are immobile? Our hypothesis. If a plant is placed in a location with this light source coming from a specific direction, then the curvature of the plant will bend towards the source at an angle of 70 degrees, and with more vertical growth compared to the group with full light exposure. Additionally, if grown in an environment that only a portion of the test source is exposed to light, then the growth of the plant will be at an angle towards the direction of the light. Instead of growing vertically, this group will result in a change in curvature to favor the portion of the environment exposed to light. This plant will not grow vertically because of the effect of phototropism. This movement is due to the circadian rhythms coordinating with the positioning of light, in some sense creating a biological clock. Thus, the cell signal processing of the plant will make the test subject move towards the light source. Experimental design. In this study, there will be two groups, one experimental and one control. The independent variable is the direction of the light source and the exposure to light relative to the box they are planted in. This will be manipulated by full exposure versus limited exposure to light. One group receiving light perpendicular to the ground, fully exposed, which will be our control group, acts as a basis for comparison to the experimental group. The experimental group is the group being placed partially in light, with a hole cut in the box. The dependent variable is the pattern in plant growth and curvature, which is measured in qualitative observations, and the degree of stem growth for both the control and experimental group.
The experimental design included the constant variables, which was the type of plants, the same species, time exposed to light, amount of water that the plant receives, the location of the experiment, the type of soil, the amount of seeds, and the time planted. For each trial, there was one done per each group. The materials included brown lentil seeds, 70 in total, organic soil, 1 cup, water, a large corrugated cartoon box, small containers, large knife, protractor, easy flex ruler, and a permanent marker. The procedures included first to bring the necessary materials out, to put the seeds in water, in a cup of water, before the day that you plant them to make sure that they start germinating at a much faster rate than not putting them in water. The second thing is cutting holes in the trays or in the pots that you are going to plant them in to make sure that the water stays at an optimum level, to make sure that if you put too much water, then the water is just going to go through the soil and then outside. And if you put too little, then you could check since none of the water is going to be dripping from down there. Now, third, place the seeds over each pot carefully over the top of the soil, and then take the fiber calm trowel and place about 1 gram or soil on top of each pot to make sure that the seeds are not completely submerged, but you just want them to be a little bit submerged, so that way it doesn't take too much time for them to grow out. For fourth, make sure to water each plant with 1 to 3 millimeters of water the first day, and then set the plants outside. And then for the rest of it, you have to use the method of figuring out how much water does each plant needs to be water, or how much water does it need, because the fixed amount is not going to work. Check the temperature outside. If it's too hot, or if you feel like you touch the soil with your hand and then you feel like it's pretty much dry, then make sure to water it sufficiently. Make sure to not water it too much or too little, using the method of that. Fifth, at day 6, randomly assign the containers into the experimental group and the control group, so that way you can get rid of confounding variables. And then bring the box, the corrugated box, outside, and cut it from the right side with a medium-sized hole that allows for some light to come into the box. You can do it from the left side depending on where do you want the light to come from. And then record the data, and for step number 6, make sure that you do the previous steps, and make sure that you measure them using the ruler and a protractor for the angle. The data, so this summarizes the data. The data was collected at day 7, and the reason why is because the box was installed on day 6, and measuring the data before it isn't going to really produce any viable results. And the reason why is because the data before both the experimental and the control group, before they were randomly assigned at day 6, were actually put in the same location, and so measuring the data there is not going to produce any significant difference, but it could have helped the experiment. In terms of analysis, it started by the chi-squared. We did chi-squared between experimental group, as you see AAC and AAE for the experimental group and the control group at the top. And what the result was 29.84, and this was bigger than the p-value of 3.84, which meant that the null hypothesis was rejected, which meant that that the experimental results are not due to random chance, that the difference between the experimental and the control are not due to random chance, but in matter of fact, they are due to the independent variable, the specific direction of light. But that doesn't mean that there's no confounding variables in the experiment. For the analysis, as you can see, there's the graph angle degrees versus time in days, and the length of the stem versus time in days. For the angle, as you see, for the control group, which was the red, and the experimental, which is the blue, the experimental has a constant decrease in terms of the angle in degrees, and the control has pretty much stays consistently the same. And the length of the stem, both of them are growing, but the experimental is growing at a much faster rate than the control is. And as you can see that, one thing that I want to highlight from this slide is the fact that these plants are competing for the same resource. These plants are, matter of fact, competing for the same resource, but in the same way they're trying to avoid it because of the law of competitive exclusion. They don't want to compete because they know that those who have the most oxygens, or those who grow the farthest, will be able to reach the most sunlight, and those who don't will be able to become extinct. So they want to grow strategically. They want to grow into sites where the resource is not being utilized by anything else except themselves, so that way they do not compete with anything else.
As we began evaluating our results and data we had collected, we can say that our hypothesis was supported due to the fact that 69.883 or 70% was the average angle of the experimental group exposed to limited sunlight. Not only this, but we can also see that an even minor error in measuring the angles could have caused data to be off, as well as not having a longer time period to conduct this experiment. And of course, a way to really improve this is to use precise measurements when it came to the angles, as well as having longer time to conduct this experiment, which will help since we could have also done more.
Before I start the communications part, what you are about to hear are two connections between the podcast and their overall message. So for the Smarty Plants podcast, it connects to our experiment due to the same overarching concept of training plants to adapt under certain circumstances when given or removed certain variables. For example, the plants grown in Smarty Plants were dropped to see if they learned the job was not an imminent threat, but rather an event that they were set upon, and due to conditioning, they learned that they in fact did not need to close their leaves when being dropped for about 60 times. They were classically conditioned to not close their leaves anymore even 28 days after the experiment, which with our lab, we removed the variable which was the full exposure to sunlight, but not just the light. It was how much access they had to it and what direction it was coming from. We conditioned the plants to adapt and change their curvature towards a specific direction of sunlight that came in from the box. This allowed them to learn and predict where the light is coming from every day, which is why they create an angle or curvature towards the sun for their own survival and benefits, which is also what the experimental plants being conditioned with the bell showed. This associative learning came into play when food, a fan, and a light were present when a bell rang. After some days, they tried to turn on the fan to see if they knew when the light was coming, and sure enough, the plants stretched their leaves towards the light, expecting it to be turned on. Same way, being that they were conditioned with the box, doing so with their circadian rhythms. For the podcast From Tree to Shiny Tree, we learned that trees within the same environment along with fungi need each other to live. This was learned throughout the experiment, which the lady Suzanne Simard took out a birch tree to see if the Douglas fir would survive or die with the absence of the other tree. It turned out that all trees need each other as they have an underground existing economy with trees and fungi. This creates a symbiotic relationship between the trees and fungi in two ways. The trees use the fungi as a storing system for the sugar and as a messenger system to the other trees. The trees use the fungi as a storing system for the sugar and as a messenger system to the other trees. The fungi then use the tree as a supply for sugar from the atmospheric carbon which they need for growth development. This connects to our experiment as our plants most likely than not created an interconnected system underground between rows to survive and thrive with each other as it is shown in the pictures and data.
For the post-lab questions, I want to go over some brief experiment that went on. In 1926, a Dutch psychologist by the name of Wens made an experiment by removing the tips of old seedlings. He put them on agar, and after the oxen has went into the agar, that agar was placed on top of the decapitated stem. And the point of this experiment is to show that the oxen really does affect the growth of the plant and really plays a big role in terms of that. And this produced the acid growth hypothesis, which provides an explanation to how oxygen causes cell wall expansion. Oxygen causes responsive cells to actively transport hydrogen atoms or ions between the cytoplasm into the cell wall. And you can see them right here. The oxygen comes in at the receptor, it stimulates it. As a result, this pumps the hydrogen ion into the cell wall, which loosens up the cellulose and reduces the pH for the enzyme to work. And the enzyme basically breaks down those cellulose, and as a result, you have the cell elongation from the auxin, and it's really due to auxin. Now, the hormones that are responsible for this is auxin, or the most common one is named IAA. Other ones are gibberellins and cytokinins. Auxin is responsible for stem elongation and the promotion of cell division. Auxin is mostly found in apical meristems, which is basically at the top of the stems. And the reason why is because auxin is trying to grow the plant upward and is trying to elongate it. That's why you don't see it at the bottom or in the middle, unless the stem is coming out. Now, the hormone auxin, and the reason why it's going to the dark position is because it can be photooxidized. And what that means is that it can be essentially destroyed in some sense, is in that it doesn't function in the optimum way that it should in the light side. So that's why it goes to the dark side, and as a result, the cells on the dark side elongate, and essentially the plants bends toward the light. And other hormones that are responsible is cytokinins, which promotes chloroplast development and assist in cell division. It stimulates it.